We're live, and welcome to another episode of Machine Medicine's interview series. And we're delighted to have Cameron McIntyre here. Thanks for being here, Cameron. And perhaps you'd kick us off with giving us a very brief bio of how you got into this field, what it was that first attracted to you, and how you finally ended up committing.
Yeah. Hi. Thanks for the opportunity to talk today. So I'm currently a professor of biomedical engineering at Case Western Reserve University. But in 2 more weeks, I'll be a professor of biomedical engineering and neurosurgery at Duke University. So kind of caught me at a transition point here. But yeah, I started in the field of biomedical engineering, I guess, as an undergraduate student at Case Western Reserve University, and didn't really know what it meant. It just sounded cool, and I was good at math and science, and so yeah, that sounds like a good idea. And along that line, or along that process, I found specifically neuroengineering, and as an undergraduate, started working, on a research lab that was actually Warren Grill's lab. He was not even a professor at the time. He was a glorified postdoc, and but starting a lab at Case Western Reserve University, and needed some warm bodies to fill some seats, and that's how we kind of met and started working. And from the get-go, we were working on electrical stimulation of the spinal cord at the time. And, we started building some computer models of how electric fields interact with neurons. And that then, became the focus of my dissertation. And then that evolved into, kind of finding an application, which was clinical deep brain stimulation. So I felt like there was a pretty substantial need to better understand how electric fields interact with, brain tissue in a human context, and deep brain stimulation was, really getting going at that point. Now we're at now we're in the early 2000s, I guess you'd say. And that's been a great, opportunity for me then over the last 20 years or so to, really focus on deep brain stimulation, building imaging and computer modeling techniques that can be applied to, kind of clinical deep brain stimulation analyses. And that's really now what our lab is focused on. So the kind of the neurosurgical planning of, brain stimulation therapies, and then the post-operative, optimization of those therapies. So I'd say those are the kind of things that we work on.
Yeah, which and very exciting work it is you're doing as well. But then, so let me be devil's advocate a bit, and then say, so are you essentially a frustrated neurosurgeon? If you had your time again, would you do neurosurgery, or does this speak to a deeper need for the interdisciplinary requirements for this work to really sort of ultimately come to fruition?
Yeah. So I never had any interest in being a clinician of any kind, neurologist or neurosurgeon. I respect you guys or gals, but it's not my cup of tea. So my motivation all along has been, I have a scientific skill or a scientific knowledge base, and I've always had a passion for trying to figure out how do you get that knowledge base out of this small little world of people that, that work on this kind of stuff, and into a bigger world of people that could benefit from it, but they've never had that training before. So, in my opinion, the whole field of neuromodulation is, pretty much populated by clinician scientists, and now becoming more widespread. And so now even more just general clinicians. But most of those people, in fact, I'd say nearly none of those people have had any real training in, kind of engineering that goes into these devices, the electrical engineering side of it, the physics of it, or more specifically to me, the biophysics of how these, kind of devices interact with the nervous system. And so I've always had that desire to figure out how do you take that kind of scientific knowledge and put it into a context that could actually be clinically useful. I don't know if we've succeeded. We're trying. I think we're getting better all the time. It's a tough nut to crack, but someone's got to do it, and I think that it needs real engineering skill. That you just don't get in medical school. So.
Yeah, but would you say that this field is sort of more in need of those engineering skills than other ones? Let's say, take just plucking out of thin air something like hematological medicine or something. It seems there are basic scientists working in hematology as well as many clinicians, but the kind of clinician scientist model seems to work pretty well. They go into the lab, they bring it straight out in clinical trials. There's been great progress in, for example, sort of hematological malignancies with that. But what is it about this area that demands a sort of greater buy-in from those hard sciences, the engineering, the biophysics that you mentioned?
Yeah, I mean, it's just, this is a very device-based clinical world, and the world of neuromodulation is just very device-based, and I don't see that changing anytime soon. And those devices are amazingly complex. I mean, all this stuff, all this electrical engineering detail, all of this, kind of really, in many ways, I think a lot of clinicians look at neuromodulation as like a black box technology. And because in medical school, they had no access to, Kirchhoff's current laws and things like that. That's not what's getting taught in medical school, right? They're getting taught drug dynamics and things of that nature, which is great because most of their real life is pharmacology. But if the world does start to transition to, being more acceptive of electrical therapies, maybe medical schools will start training them more on the electrical side of how, kind of information transfer can happen in the brain or in the peripheral nervous system or whatever. But until that happens, you're very much dependent on these devices to just work. And it would probably be better if you actually understood how they worked kind of, let's call it at the neural level or at the neurotransmitter level. And so it's a the time constant of change is very, very slow in medicine. Because the training paradigms, are very slow to change, and then the residency training is very slow to change, and then the practice patterns are very slow. So it takes a long time, and until we get to that point, I think there is a great need for let's call it biomedical engineering type people, to kind of play that intermediate role, and that's what I try to train people, to be those kinds of, scientists, clinician or clinical scientists. But it's a slow process. So.
I couldn't agree more. And I can tell you from firsthand experience, having gone through medical school in Oxford and tried all through that time to get sort of quantitative training alongside clinical training and biological training, that it was very, very challenging indeed. I did manage to get my college to pay for sort of maths tutorials alongside my sort of biological and clinical studies, but it was really an uphill struggle, and let's hope that's changing. But it's sort of I think I'm totally convinced that you're right, I think. But one of the things I think sort of slightly curious, and perhaps you could give us the benefit of your thoughts on why this is that actually arguably kind of neuromodulation, quote unquote, has already taken place about 30 or 40 years ahead of us in the context of cardiac medicine. So, which is really a very similar technology, right, with pacing, and yet in this field, it seemed to happen in like the 70s and 80s, and is now is really a very mature kind of clinical. So, what's different about the excitable tissue in the heart and what we're trying to do in neuromodulation? That means there's nigh on half a century between sort of progress.
Yeah. I mean, the fundamentals are exactly the same. You're just using an electric field to manipulate the voltage sensor and a sodium channel. That's it. That's it. Okay. Like at the basics, it doesn't matter whether that's in the heart or in the brain.
Yeah.
I think where the big jump happens though, or why. So, I don't know anything about the cardiac world, and maybe cardiologists would feel like, hey, we understand this and we know what we're doing, and we don't need, engineers to help us use these devices efficiently or effectively, or, whatever. They're optimized and they work great. And I don't want anyone to take offense to this, but, the heart's just a much, much, much more simple system than the brain or the peripheral nervous system for that matter, either way, right?
Yeah.
Like you have, I don't know, let's say 10 different neurotransmitter systems that you are potentially, manipulating. And you've got literally millions of different circuits that you are manipulating with, electrodes implanted in the nervous system. And the heart is kind of, maybe people who are experts in that would say different, but it's, in my opinion, it's like it's one circuit, it's one system that you are, pulsing. And so, just a much more approachable problem. And that's why it worked first. And that's why it, was, I think, very successful, even in a time of maybe relatively limited scientific and engineering, kind of like detail, but was able to work very efficiently and effectively in a clinical environment. Brain neuromodulation is, that's the biggest, unsolved, like machine in the universe, right? We are barely scratching the surface, and now you throw in, your manipulation of it with electric fields, and that gets complicated real fast.
So yeah, and that's another reason why I think it's interesting. So you mentioned the fact that just like kind of the state space that you're dealing with a cardiac, with a heart and versus a central nervous system is just like orders of magnitude apart. Albeit the state space for the heart may well be very complex and high dimensional itself. But one of the other interesting things is that at least in cardiac medicine, there seems to have been a great deal of success with recording an ECG, or with you guys EKG, and then using that as essentially a proxy for cardiac function, and then feeding that back into the system, optimizing the parameters. And actually, with an EKG or an ECG, you actually can do a pretty good job. It's a very the correlation coefficient between the EKG and cardiac function is pretty damn high, so you can get a lot of good clinical information essentially about function from that. And there seems to be, there's a lot of excitement now in the field, what with things like Percept systems coming out at a commercial kind of stage of development, that we're going to be able to do something very similar in the brain with essentially just sensing things like beta rhythms and stuff. And perhaps this will bring us on to some of your other work where you're sort of integrating different modalities. But what are your thoughts on that analogy? Is that an oversimplification?
Oh no, I think that's exactly where we're going. We've been attempting to talk to the nervous system with our electrical pulses, for whatever, let's say, 30, 40, 50 years. And, it's been a one-sided conversation, and we don't know if what we're trying to say is really getting through or not. We know if we get the behavioral effect, then all right, you heard what we were saying and you did it. But clearly, it would be a lot more elegant if we had a better understanding of the language and we could give them information, right, in a way that the nervous system could interpret better, and we could do that presumably a lot more efficiently if we understood what it was trying to tell us back. And that's the whole goal of these kind of LFP, and really the whole field of brain-machine interfacing. How do we listen and respond, appropriately with our stimulation? And maybe LFPs are the way to go, or maybe not. I don't know. We'll figure it out. So.
Yeah, let me push you on that a little bit more, and bring you back to your comments on the complexity of the system. So as we mentioned, you can essentially record at one site in the heart and get a decent proxy for global function. But if we're recording at one or even two or three sites in the brain, is it really plausible that we could get a global readout of kind of neuronal function?
Definitely not a global one. If it was that simple, we would have been able to solve this problem with EEG, 50 years ago, right? And we haven't. So, But I think the goal is, maybe you don't need a global readout. You just need a local readout that is robust and reproducible, and that you can rely on to trigger your stimulation. And it's a proxy. It doesn't have to be, you don't have to understand the actual, language of the brain or the science or whatever. All you need is a control signal. So from an engineering perspective, right? Let's not make it a science project if it doesn't need to be one. And I think that, that's where we're starting right now. And no, I mean, in my opinion, no, we don't really know the best place to put the recording electrode, to make a really efficient closed loop deep brain stimulation system. But you have to have the tools to start testing, different ideas. And that was what I think was, a great thing that happened with the Activa PC plus S, whatever, 5-10 years ago, was you start putting this technology in the hands of creative clinician scientists, and they're gonna test some cool ideas, and that's how you'll figure out where to put the electrodes, once you get to the point where you're ready for a real closed loop device. And I'd say we're only at the early stages of that process now. So.
Yeah. But it's interesting to hear you sort of evince such faith in the possibility of just using a control signal, especially in regard to, do you think the rise of machine learning over the last sort of 10 years, certainly in terms of sort of prominence, if not sort of in terms of and computational power, is that the sort of, do you think that's going to be a big contributor to, taking these control signals and actually learning in the data-driven way?
Oh, well, I mean, that is certainly the hot topic right now. I'm personally not sold on it, but that doesn't mean that it's not going to work. I've been wrong a lot more times than I've been right. So, I think that is definitely one approach to, kind of trying to map out this system. Like we don't know the language, and so we're trying to figure out, where should we record and how should we interpret those signals, and machine learning is a massively powerful tool to attempt that. But one of the things that I feel like is, machine learning works awesome when you're trying to characterize a stable system and a reproducible system. But we don't really know whether most of these signals, one, we don't really know where to record, and two, we don't know the stability, and three, we don't really know the dynamics of them, and so we're kind of throwing black box algorithms at stuff, hoping we get something that will be, a robust way to predict or give us a control signal. But, that's still, I think the jury's still out on that, to me. So. Yeah,
absolutely. And maybe there's a strong argument there that actually what we need also is clinical feedback.
Yes.
So actual sort of functional sort of readouts of how clinically well the patient's recording in conjunction with those internal signals and many others. But this again sort of, brings us on to sort of interesting work that you've been doing with sort of multimodal sort of integration and some of the challenges associated with that. In particular, I was having a look at your paper on StimVision V2. So this is a really cool sort of platform, as you describe it, and perhaps you'd like to say a couple of words about the motivation behind it first, and then sort of what it does and some of the challenges that you faced, and how it maybe illustrates some of the challenges for the field in general.
Yeah. So our work, or our lab has spent, many years trying to develop software technologies that integrate brain imaging data with biophysical computational modeling, specifically in, the subthalamic region for the treatment of Parkinson's disease, but also, in the subcolossal cingulate region for the treatment of, experimental treatment of depression, and, a variety of other, kind of deep brain stimulation research, kind of arenas. And, I feel like we have always struggled with, kind of how much can we learn from a single patient versus how much can we learn from a population of subjects kind of, looked at, smeared together. And so there's kind of 2 or 3 different philosophies on, what the best approach is and how can you build these, we call them structure function relationships, right? We would like to know the specific neural, what in your definition of whatever that is, whether it's a nucleus or a fiber pathway or a neuron type, whatever it is, some neural, response that is associated with some behavioral output.
Yeah.
And if you stimulate it, you stimulate X, you get behavior Y. And that is the basis of, I would say, kind of brain science for 100 years, I guess. And as we've kind of evolved, like whatever, 30, 40 years ago, we're like, okay, the subthalamic nucleus is the target that we are stimulating for therapeutic benefit for Parkinson's disease. And then, people started thinking, well, you know what, maybe it's actually all these other, axonal pathways that are streaming around the subthalamic nucleus. That maybe we're stimulating those, and that's what's giving us benefit. And the subthalamic nucleus is really just a targeting proxy. That it's not necessarily the thing that we're stimulating for benefit. And so let's say that was kind of the scientific hypothesis, that started coming together in the last 10 or so years. But frankly, in my opinion, we're still, that is still an ongoing debate. We don't really know, is it the subthalamic nucleus neurons? Is it these axonal fiber pathways passing above the subthalamic nucleus? So we're still trying to figure out that question. And that would be interesting or important to answer that question, because it would help us, one, with targeting of the electrodes, right? How do you customize the targeting to a specific patient? And it'd be helpful from a scientific perspective, because if we knew what the real target was, we could presumably design, the stimulation to better, to better or more efficiently stimulate that target. So anyway, so that's really what, kind of the software technologies that I'm interested in building, or that our lab builds, are really focused on, is how can we kind of answer those questions, and what is the right kind of imaging data that you need? And then what is the right kind of computational method to analyze that imaging data with? And then what is the right kind of, I'd say, biophysical model to use to try and, get at some of these different hypotheses. So yeah, that's kind of general. I don't know if you want more specifics than that, but that's, kind of where we're going. And I'd like to actually bring it back to the point that you made earlier, which is you can do a whole lot of stuff with building computer models and pretty pictures and MRI data sets. And, that's very popular right now in the DBS world. And, we're learning some stuff from that. But the only thing that really matters is, what comes out on the behavioral output side, because that's the only thing that matters. Who cares if we actually understand, the details? And that's where, let's say the brain-machine interfacing side of research and deep brain stimulation is very myopically focused on, electrophysiology and local field potentials and whatnot. But, we should probably be spending more time on, kind of like the peripheral side of that recording and monitoring and documentation, because, that's a lot closer to the real thing that we're trying to manipulate than the local field potential is. So just to bring it back to that point you were saying before of, hey, what about some of the behavioral stuff, and can we use that in these kinds of closed loop systems? Yeah, I think we probably should be doing more of that.
Yeah. And so with the STIM Vision 2 work, one of the things when I was reading the paper that I was struck by was actually how much of the detail was just about kind of putting things into sort of co-register, kind of data compatibility, these kind of, and it really speaks to this issue that we have about sort of the lack of interoperability. and how it's a hindrance everywhere in medicine, but here it just seems to be almost like 90% of the problem in almost everything anyone does. Is that fair characterization?
Unfortunately, I think it is. I mean, there's so many steps, and so part of it is that the questions that we're asking are, really complex now. We're past most of the easy questions. And so when we're trying to dissect the mechanisms of deep brain stimulation, we're now trying to ask, or we're trying to answer questions that are really, pretty detailed. And so now, the nuances of, 1 millimeter, it like can make a big difference in your conclusion. And so, when we're trying to use imaging data that at best is giving us something that's on the resolution of 1 millimeter, now, like you're running up against this thing where how you're doing your data registration is actually just as important as, the methods and the hypotheses that, you're crafting. And so, can we answer these questions? I mean, I think we're right at that limit. We're trying to ask questions that are right at the limit of what the imaging, data might possibly be able to give us, information on. And to me, that's what a model is for, though. Like you've got obvious things that you can see, and then you build a model that helps you, kind of take what you can easily see and just get your knowledge base, 1 or 2 steps further than that. And then that sets up the motivation for, okay, here is the next big thing that we really need, from the imaging world or from the behavioral outcomes measurement world or whatever. So that's where I hope that, we all are working, kind of together with that, because you need the imaging world to improve. You need the modeling world to improve, and you need the behavior outcomes measurement world to improve if you're ever actually going to answer any of these questions. So.
Yeah. And how about kind of slightly more arcane areas like the field of computational neuroscience and trying to understand things like, the neural coding? Are these things relevant yet for building these kind of systems, or is it still slightly kind of an occupation for slightly self-indulgent physicists?
So so in 2002, the first grant I ever wrote was that I was going to build a network model of the thalamocortical basal ganglia system, and I was going to understand the mechanisms of deep brain stimulation, right? Okay, so that's how I started, and the more I learned, the deeper I got. 20 years later, I'm like, oh my God, we're so far away from being able to do that. It's like impossible. Okay. Now that's just my feeling. A lot of people right now are doing pretty cool stuff, looking at network models and trying to couple those network models with, the brain imaging data and whatnot. And, I'm sure they're going to see cool stuff. And maybe that really is something that's ready for prime time. To me, like there's so many unknown parameters in a giant network model like that. It's hard for me to, to really sink my teeth into it and really believe it. But, you have to try and then figure out where the model breaks down and then, see if you can fix it. So I think we're at the verge of people really successfully, attempting those kinds of things. And, but it's just not my personal, kind of direction. Cause I like things that I can really like, grab and say, okay, I know what this parameter is. I can quantify it. I can measure it, and I can document it. Yeah.
Yeah. So maybe would it be fair to kind of to think that perhaps there's going to be at a long time scale, a kind of 2-step process, one where we're kind of blindly developing kind of model-free, empirically driven sort of systems that work. And then subsequently, as we get more learning, we'll ultimately be able to build, I guess what we hope, what we all hope will ultimately be possible one day, a kind of, silicon double of in silico double of a of a patient that allows us to perfectly mechanistically understand both their sort of their disease state and how an intervention will affect that, and that, who knows, that might be 15, 20 years down the line. But in the interim, there's a lot we can do with an empirically driven kind of pragmatic approach. Would that be fair?
Absolutely. And that's where, like half of my life, I think of myself as an engineer. Yeah. And another half of my life, I think of myself as a computational neuroscientist, I guess you'd say.
Yeah.
And like I'm probably more motivated by the engineering side. And like, let's solve the real goal, which is let's make this device, be as effective as it possibly can be. And the way you're going to do that, is with, like the first model that you talked about, we don't have to understand the mechanisms to optimize it, to some extent. But if you really want to, truly make it a great, interface with the patient, then yes, eventually someday you need to actually understand the real network and understand the real language of that circuit. But you can, there are intermediate steps, and let's take advantage of those engineering steps that don't necessarily need, the documented basic science at every level. So
yeah, cool. And there's one more. I'm conscious of time, but there's one more question I would really like to ask you and hear your thoughts on, which is slightly sort of oblique but related to what we've just been discussing, and that's the issue of sort of hardware versus software. Now, I think you can make a fairly sort of strong argument that today the field is hardware dominance, and people like Medtronic and Abbott and so forth that build these devices typically have their hardware engineers first and foremost. But perhaps by analogy with sort of general computing, there may be a shift in the future from the true kind of value and the real sort of interesting developments happening on the hardware side to those developments actually sort of shifting up to the software side, and ultimately neuromodulation being actually a software game first and foremost, rather than a hardware one. What are your thoughts on that?
Yeah, I sure hope so. That'd be good for my career. So, but no, absolutely. That's why I, trust me, when I was in graduate school, I wanted to build, build an IPG or whatever, right? That's a tangible thing. Like, that's awesome. Like, here is a device as an engineer that you built, and that is so cool. But I guess I came to a realization that the opportunity for impact, at least for an academic researcher, was really on the software side. And an academic type person is never going to have the infrastructure to build a competitive clinical level neuromodulation device, right? I mean, there's just realities that prohibit that kind of thing from happening at the scale that would be necessary, and so we have a lot of different devices that are out there and at different levels, right? There's kind of research things all the way to kind of very, very simple neuromodulation devices, but somewhere along the line we will start to customize them to individual disease states. And have, real engineering criteria that say, okay, this disease state, we really need to have, 15 channels and 3 different leads placed in these specific locations. We're a long way from that now, but we will eventually get there. I do believe that. And once you get to the point where you have, I think, come up with a relatively customized therapy, hardware therapy for that disorder, then the real differentiator will be the software integration of how you are communicating with the nervous system via that hardware. And, well, right now I'd say we're only beginning to learn how to do that. But, once you know what the hardware requirements are, it's pretty easy to get those kind of devices built. The differentiator will be the software, and that's what will make a big, kind of impact, and whether company A will be able to, offer the coolest features compared to company B. And and not just, from a competitive perspective, but from a patient outcome perspective, like how do you customize it to that patient and customize it to the specific percentage of therapy or percentage of symptoms that one patient has, right? Like for example, in Parkinson's disease, you've got tremor dominant patients and you've got, kind of rigidity dominant patients. And my guess is that you probably want to stimulate those two different patient populations in different ways. And the software is what's going to figure that out for you. The hardware is going to just probably be the same, for each of those different applications. So yeah, I think it's, I think that that's the future. And that's why I've, spent my career working on it. So
cool. Well, look, Cameron, I was looking forward to chatting to you today, and it's been a pleasure as I anticipated. And we'll be keeping an eye on your work in machine medicine, and hopefully bump into you at one of the neuromodulation conferences if they ever actually do start taking place physically again. But yeah, thanks again for taking the time to chat to us, and look forward to catching up again soon.
Absolutely. I had fun. Thanks for the opportunity. So.
Cheers.
Okay.